Check, 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 check. Very good evening, children. So we'll start the class. If you all join, we'll start the class, children. Welcome to today's class. Very good evening, children. We'll start our today's class. The today's class topic is about biotechnology and its applications. Biotechnology and its applications. So let me define you what is biotechnology, and then I'll explain about the history of biotechnology. Biotechnology, it is a science of biological process where the biological organisms are involved in for the benefit of mankind. Here the process, developmental process or utilization of the biological process mainly for the benefit of human being or life form, it is called as biotechnology. It is using the biological organism for the benefit of humankind. That is the ultimate aim of biotechnology. The term biotechnology, it was coined by Karl Erke, an Hungarian engineer, in the year 1919. And here this biotechnology, any process that involves an organism, a cell, a tissue, an organ system, or organelles or molecules or an enzyme, if anything is involved for the benefit of humankind, that is called as biotechnology. It includes any process, an enzyme, the production of an enzyme, production of secondary metabolites, production of genetically modified organisms, any raw material for the humankind, it is called as biotechnology. So let us learn about the history of biotechnology. The history of biotechnology. In the year 1770, Anthony Lavoisier, he gave the chemical basis of alcoholic fermentation. He is the first person who identified the fermentation process in the case of alcohol. And after that, Edward Jenner, he introduced the first viral vaccine to inoculate in the child who suffered with smallpox. And in the year 1838, the protein discovery was made, and it was recorded by Gerardus Johann Mulder and Johannes Jacob Berlisius. And in the year 1876, Louis Pasteur has identified the role of microorganisms in the process of fermentation. And in the year 1919, the term biotechnology was coined by Karl Erke. And in the year 1928, the discovery of penicillin, it was done by Alexander Fleming. And in the year 1941, Beadle and Tatum, they have identified and gave the concept one gene, one enzyme hypothesis when we are doing the experiment Neurospora crassa. And in the year 1944, Avery, McCall and McCarthy, they have identified that DNA is the genetic material that is present in the organisms after doing lot of experiments. And in the year 1953, the double helical model of DNA, it was identified by James Watson and Francis Crick. And in 1972, the discovery of restriction enzymes, the enzymes that are involved in genetic engineering process, were identified by Arber, Smith and Nathans. In 1973, the fragmentation of DNA, and they combined with plasmid vectors. So that is the recombinant DNA technology. It was identified by Stanley Cohen, Annie Chong and Robert Helling and Herbert Boyer. And 1975, in the year 1975, production of the monoclonal antibodies from clone cells were identified by Kohler and Milston. And in the year 1976, Sanger and Gilbert, they have introduced the techniques, various techniques to sequence the DNA which is present in the chromosomes. In 1978, production of human insulin in E. coli cells were identified. And in 1979, development of artificial genes was isolated and it was developed by H.G. Corona. And in the year 1982, the first human insulin, that is called Humulin, this is the first pharmaceutical product of recombinant DNA technology. It was approved by the government of the United States and it has come to the human use. And 1983, the tumor inducing plasmids, TA plasmids, it was introduced in the field of for transferring the genes in the field of genetic engineering. In 1986, the PCR, the polymerase chain reaction, which are used to manipulate, which are used to produce multiple copies of DNA, it was identified by Carl Mullis. 1987, the gene transfer, one of the method, it is called biolistic method, that method was introduced. In 1992, the first chromosomes of yeast, it was sequenced. And 1994, the genetically modified flavor savage tomato, this was introduced, this was approved by the United States of America government. In 1996-97, during that year, the first transgenic animal, the mammalian sheep dolly, was produced by Dr. Ian Wilmot in Scotland. And in the year 2000, the first plant genome of Arabidopsis thaliana, it was sequenced. And 2001, the Human Genome Project creates a draft of human sequences. And 2001, the Human Genome Project. What is Human Genome Project? The study about the complete sequence of DNA in the human beings. It was started, and 2002, the first crop plant genome, it was sequenced in Oryza sativa plant. And 2003, the Human Genome Project, it get completed, and they have identified that they have sequenced all the human genes of all 46 chromosomes. And 2010, Sir Robert Edwards, he developed the in vitro fertilization process in the case of animals. In 2016, the stem cells were injected into the stroke patients and enable the patient to walk. This is the stem cell therapy. It was introduced in the year 2016. 2017, the blood stem cells grown in laboratory conditions. In 2018, James Allison and Tasuku Honja, they have discovered protein found in human cells or immune cells, and their role in cancer therapy, it was identified. Next, we are going to talk about the development of biotechnology. The biotechnology, we can categorize them and study as two things. One is called traditional biotechnology as well as modern biotechnology. Traditional biotechnology is also known as the conventional biotechnology. So, the biotechnological process, it was not just started few years back or few decades back. It was long back. It has been a practice with us. For example, the use of bacteria and other microbes in daily life is a classical example of traditional biotechnology. The production of dairy products like curd, ghee, cheese, and preparation of food like batter, preparation for idli, dosa, naan, bread and pizzas are example for that. And also the beverages, the beer and wine, these are all the traditional method of biotechnological process. The modern biotechnology, there are two main features are there in this modern biotechnological process. One is ability to change the genetic material for getting new products through recombinant DNA technology. Getting a new product, that is one of the important features of modern biotechnology. And second thing is ownership of newly developed technology and its social impact. So one is production of new product, other one is how to manage the product. These are the two main features in the modern biotechnology. So there are several improvement it was there in biotechnology. Various fields were developed. Mainly the biotechnological products are dominating in the field of pharmaceutical companies, beverages, agro based industries, and also various biotechnological field tools were developed for the improvement and betterment of human life. The modern biotechnology, it includes all methods of genetic modifications by using the recombinant DNA technology and cell fusion technology or protoplasmic fusion technology. What are the main focus of this modern biotechnology? It is fermentation of acids, enzymes, alcohols, antibodies, antibiotics, chemicals, vitamins and toxins. Next is biomass for bulk production of single cell proteins, alcohols and biofuels, and enzymes as biosensors, and biofuels for the production of hydrogen, alcohol and methane, and microbial inoculants as biofertilizers and nitrofixes, and plant and animal cell culture mainly for the production of secondary metabolites and monoclonal antibodies. Recombinant DNA technology for the production of enzymes, vaccine, growth hormones, secondary metabolites, antibiotics as well as interferons, antiviral proteins. And process engineering, mainly this process engineering, it deals with handling the effluent, the waste produced from the sewage as well as the recycling of water. These are the important modern biotechnological focuses. Now the important thing in biotechnology, we are going to talk about genetic engineering process. Genetic engineering process. The genetic engineering process, the two core techniques are there in modern biotechnology. One is the genetic engineering and the other one is the manufacturing of antibodies, vaccines and drugs by using genetic engineering concepts. So what is the genetic engineering? Here, this is the manipulation of the genome of the organisms. We are altering the genome of the organisms and transferring the desired gene into the organism mainly to improve a particular function or a particular trait. If suppose there is an abnormal gene is there in an organism, because of that abnormal gene, the organism could not able to produce a particular enzyme or a particular trait or character. So that what we can do, we can manipulate, we can alter the genome so that what will happen, the genes get altered and that particular trait or particular character, a particular trait, a particular character, particular enzymes can be altered. That is the ultimate aim of genetic engineering process. And also genetic engineering, it involves the manufacturing of antibodies, antibiotics, vaccines and drugs for humankind. Now let us see about how this manipulation of genome it was done in the field of genetic engineering. The genetic engineering is also known as recombinant DNA technology, or it is also known as gene cloning. What we are actually doing it is, here we are modifying or manipulating the DNA from one organism to another. Manipulating the DNA from one organism to another, or transferring a gene from one organism to another. That is the ultimate aim of genetic engineering or recombinant DNA technology. So now let us learn about what are the tools of genetic engineering. Now we know what is genetic engineering. It is the manipulation of the DNA by altering the genes. And this tools of genetic engineering, it includes enzymes, vectors and host organisms. These are the three important tools of genetic engineering. Among these, the most important enzymes that are involved in genetic engineering are restriction enzymes. The restriction enzymes, we have restriction endonuclease, restriction exonucleases, and then DNA ligase and alkaline phosphatase. These are all the enzymes which are involved in the field of genetic engineering. Similarly, the vectors are mainly the plasmids. Vectors are organisms, vectors are the DNA molecules that are used to transfer the genes from one organism to another organism. Similarly, the host organism, it is the one which helps in multiplication of the desired gene. So these are the three important tools of genetic engineering. One is the enzymes, vectors and host organisms. Now first let us learn about the enzymes. In the enzymes, there are three important group of enzymes need to be studied in genetic engineering. One is the restriction enzymes. That is one category. The other category, it is the DNA ligase, and third category, alkaline phosphatase. The first let us learn about the restriction enzymes. So you can see this is the DNA. This is the DNA. You can see all, what is this? These are all the nitrogenous bases. So those nitrogenous bases, it has to be altered so that what will happen, a gene can be inserted, or to cut a particular sequence of DNA. The enzymes that are involved are called the restriction enzymes. The two enzymes that are mainly involved, the two group of enzymes are restriction endonuclease and restriction exonuclease. Here, these restriction enzymes are mainly used to cut the DNA at a specific site. It is used to cut the DNA at a specific site. Once the enzyme was added into the DNA, it cut the DNA at a specific site. It adds methyl group to DNA. Similarly, it cut the DNA. That's why it is called restriction enzymes or restriction endonuclease enzymes. These restriction endonuclease enzymes are involved in cleaving the DNA molecule and making the fragments of DNA at specific site or restriction sites. Everywhere or anywhere it will not cut. It will cut only at a specific site. That's why these enzymes are known as restriction enzymes. Based on the mode of action, the restriction enzymes are of two types: restriction exonucleases, restriction endonucleases. Now let me tell you what are the difference between that. Just see that this is the DNA template and this is the action of exonucleases and endonucleases. The exo means outer. This enzyme, the exonuclease enzyme, it removes the nucleotide. It cuts the nucleotide one at the time from end of the DNA molecule. You can see that the end of the DNA molecule. At the end it will cut. Whereas the endonuclease enzyme, it cut at the center. It cut at the center. It breaks this endonuclease enzyme. It breaks the internal phosphodiester bond that is present between the phosphate group with the sugar molecules. It cut that, the phosphodiester bond within the DNA molecule. So that is the function of the endonuclease enzymes. Exonucleases, it remove the nucleotide at end, from the end, whereas endonucleases, it cut at the center, mainly the phosphodiester bond within the DNA molecules. Next is some of the examples of restriction enzymes. And here you can see that there are two type of cut you can observe here. One is called the blunt cut, the other one is called sticky cut. The type of cut here Can you see this cut? This cut is called sticky cut, whereas this cut is called blend cut. For cutting a particular DNA, whether the cut has to be made sticky end or even sticky end, which particular nucleotide it has to cut, so that it can match with the new inserting DNA. Like that, different type of enzymes are involved in adding the nucleotides. You have the sticky end as well as the blunt end. For that, different type of source of enzymes are involved in that. Here we have list of enzymes. These enzymes are called restriction enzymes. One is EcoR1, that is called, it is from Escherichia coli RY13 bacteria, and HIND3, this is an enzyme which is from the Haemophilus influenza, and BAMH1, that is called Bacillus ameloleukophaciens H, and ACAL1, it is called Streptomyces albus G, and BAL1, that is called Brevibacterium albinum, and HAE3, that is called Haemophilus aegyptus, and SMA1, that is called Serratia marcescens. These are all different type of sources of and examples of enzymes, and all these enzymes except BAL1, HAE3 and SMA1, other enzymes, it cut the DNA at sticky end, whereas the three enzymes BAL1 and HAE3 as well as SMA1, it always cut the DNA at blunt ends. Now let us see about the restriction endonuclease enzyme. The restriction endonuclease enzyme, it is also known as molecular scissors. Molecular scissors. Why it is called molecular scissors? Because it cuts the DNA. You know the scissor function, right? It cuts any material. Similarly, these restriction enzymes, the endonuclease enzyme, it cuts the DNA mainly, whether it's going to be sticky end or blunt end. These restriction enzymes are mainly used to cut the DNA into fragments. That's why it is known as molecular scissors. The source of these enzymes are mainly the bacteria. And these enzymes exist in many bacteria mainly for defense mechanism. They are called restriction modification systems. There are three main classes of restriction endonuclease enzymes are present in various bacteria: type 1, type 2 and type 3. And each type 1, type 2 and type 3, they are slightly modified in the mode of action, but all of them, they are used to cut the DNA at fragment. Whereas in the case of restriction recombinant DNA technology or genetic engineering process, only type 2 enzymes are preferred in recombinant DNA technology, because they recognize and cut DNA with a specific sequence, mainly 4 to 8 base pairs. So that is why the type 2 enzymes are mainly used in genetic engineering process. And some of the type 2 enzymes that are used in the recombinant DNA technology or genetic engineering are ALU1, BAMH1, EcoR1, HAE3, HIND3. These are all some of the enzymes, and the sources are Arthobacter luteus and Bacillus ameloleukophaciens, Escherichia coli bacterium, and Haemophilus aegyptus and Haemophilus influenza. And the end ALU1, you can see that it always cut at blunt end between the guanine and cytosine. So that's the blunt end. And whereas the BAMH1, it cuts the DNA at sticky end. The cut, it will be between GATC. That type of sticky cut, it will be there. Whereas the EcoRI1, it always cut with AATT sticky end, because you know that this is one strand, this other strand, like that it will cut. Similarly, HAA3, it cuts the blunt end between adenine, guanine. Here it is guanine, cytosine cut. So that is the difference between this blunt end and this blunt end cut. Whereas here the Hind 3 it always cut at sticky end, AGCT, AGCT. Like that the cut it will be made. These are the type 2 restriction enzymes which are mainly used in the field of genetic engineering or recombinant DNA technology. And next is the naming of enzymes. We have learned that the different names are here, right? How the naming it was done. For example, there is a standard procedure is there for the naming of the organisms. Here you can see that. Let us see example. Let us take as Eco R1. Here Eco R1. The E it stands for the first genus letter E, Escherichia coli. And CO here, CO it is the species name. Coli is a species name, right? From that you take the 2 letters. So Eco, and R it refers to the strain. And here 1 means the first enzyme that is discovered in that lab. For example, here HAE3, H stands for Haemophilus, A, H starting letter, and AE, Aegyptus, AE3, that is the third enzyme identified in that particular lab. This is how the naming it will be done. Always the first letter of the genus name it will be in the first, and the 2 letters from the species name, the first 2 letters it will be added. Along with that, the enzymes, the discovery of the enzymes. If strains are there, the large strains also. For example, here BAMH, H strain, here it is R strain. So this is how the naming it will be done. The Roman letter what is here, 3, 1, 2, all those things indicating the order of discovery of the enzymes. This is how the naming it will be done. Similarly, the kind of cleavage produced by the enzymes, as I mentioned, there will be sticky ends, there will be blunt ends. Sticky end or blunt end. And some enzymes it cleaves both strands of DNA at the center, resulting in the blunt end. So the blunt end like this. This is the blunt cut. This is of course symmetrical cut. And the sticky end, the sticky cut, this is called cohesive cut, cohesive end. This is called a staggered or asymmetric cut. So this type of cut it is called asymmetric cut. This type of cut it is called symmetric cut. These are all the types of cut made by the restriction endonuclease enzymes. Next is the DNA ligase. This is an enzyme. It is called a paste enzyme. Previously, the restriction endonuclease enzymes, they are all molecular scissors. They are used to cut the DNA, whereas this particular enzyme, DNA ligase, it is helped to join the sugar and the phosphate molecule. So this one it links the phosphodiester bond between the phosphate and the sugar molecule in the presence of ATP. So here with the help of the adenosine triphosphate, the 2 strands of DNA, between the phosphate and the sugar molecule, it will be linked by the enzyme called DNA ligase enzyme. It forms the new 3-5' phosphodiester bond between the sugar molecule and the phosphate group. Next is the alkaline phosphatase. This is another enzyme. It is a DNA modifying enzymes, and this one adds or removes a specific phosphate group at 5' terminus of double stranded DNA molecule, and also single stranded DNA molecule or RNA molecule. It prevents the self ligation. That is the main function of alkaline phosphatase, and this enzyme is purified from bacteria as well as from cough intestine. So this is about the alkaline phosphatase enzyme. Next is about, we are going to talk about the vectors. In the genetic engineering process, there are 3 important things are there. One is the enzymes. So far we have learnt about the enzymes. Next is the vectors, and third one about the cloning organisms. Cloning organisms are the one. So now let us see about the vectors. So before that, I will give you a few minutes. I want you to draw this diagram. Kindly draw this diagram, and then I will come and explain you about the vectors. So we will go for a break, just 5 minutes break. We learn about the vectors. Children, next topic what I learned today is about the vectors. What are these vectors? The vectors are the vehicles, cloning vehicle, that are used to transfer the desired genes from one organism to another organisms. All these vectors are small DNA molecules which have the ability to replicate on their own. They are used as a carrier mainly to transfer the small DNA fragment from one organism to another organisms. Vectors are also called as cloning vehicle or cloning DNA molecules. Here, this is a vector which is called the plasmid vector. These plasmid vectors are taken from the bacteriums. Certain bacteriums, they have extra chromosomal materials. They have extra chromosomal materials in the cytoplasm. That extra chromosomal material in the certain bacteria are known as plasmids. These plasmids are used to transfer the desired genes from one organism to another organisms. Here, this is an gene of interest what you are supposed to transfer. So what you have to do it is, we have to take this DNA and identify the genes that has to be transferred, and with the help of the restriction enzymes we have to cut them at a specific site. Similarly, the same restriction enzymes, it is used to cut the plasmid, and after that what will happen, this DNA molecule, the gene of interest, it will be inserted inside the plasmid. Now the plasmid, it will become a recombinant plasmid. So this plasmid, now it will be inserted into the bacterium so that they can produce the same type of copies of bacteriums. That is called the manipulation or multiplication or molecular cloning process. Some of the vectors that are used in recombinant DNA technology are plasmid vectors and bacterial artificial chromosomes and yeast artificial chromosomes, expression vectors, cosmid vectors. These cosmid vectors are combination of plasmid and phage and lambda phage vector. These are all different types of vectors that are used in the field of recombinant DNA technology. Now let us learn about the types of vectors. There are basically two different types of vectors are there. One is called the cloning vector, the other one is called expression vector. The cloning vector are used to insert the desired gene from one to another, or it is used to insert the DNA into the host cell. That is the function of cloning vector. Expression vector, it is used to express the DNA insert to form or to produce a specific protein inside the host. One vector, it is just a vehicle which transfers the gene into the suitable host cells. The other one, the other vector, it is called expression vector, which are used to express the DNA insert for production of a specific protein or an enzyme or insulin or interferon, whatever it is. That is called expression vector. Now let us see about the properties of a vector. What are the important properties of the vector? All the vectors, they are supposed to do the self-replication process. They should replicate on their own. Independent replication should be there so that they can produce multiple copies of the same. And also the other important characters of vector, the vector, it should be, when it inserted inside the host cell, it should get along with the host cell and it should express the characteristics. The other thing is, it should be small in size and also low molecular weight, less than 10 kb, 10 kilo base pair, it has to be there in size. Next is, the vector must contain an origin of replication region. That is the region where it can replicate within the host cells. And also it should contain a suitable marker, that is called antibiotic resistant to permit its direction, to permit the deduction in the transformed test cells. So what should be there? The origin of replication should be there for them to replicate within the host cells, and also suitable antibiotic resistant marker, it will be there, that is to permit its deduction in transformed host cells. Reduction of any foreign substances are there, or to show resistant to certain antibiotics. And also the vector should have a unique target sites for the integration with the DNA insert. So each and every vector, just like that, it cannot combine with the genes. Even in the combining, it should identify the target sites where it has to express its characteristics, and also should have the ability to integrate with the DNA insert. It carries into the, it should carry the genome into the host cell, and also incorporate the genome what it carries into the genome of the host cells. And cloning vectors have more than one restriction site, it has to be there. It's called multiple cloning sites, or polylinker, it has to be there. These are all the properties of the vectors. And the features that are required to facilitate cloning into a vector. What are the features that are required? The features that are required are the cloning site, the origin of replication, and selectable markers. This is the typical vector. The typical vector, it should contain a region called ORI region. This is called origin of replication region. This is the region where exactly the replication process starts. This is a piece of DNA. When linked to the sequence, it can be made to replicate within the host cell. So this is the region where exactly the replication process should be there, a replication site, that is called origin of replication. Next is the selectable markers has to be there. These selectable markers, it help to identify the transformants and non-transformants. It eliminates the non-transformants, and selectively it will allow the growth of transformants. So what has to be grown, what has to be suppressed. So these selectable markers, it will identify the transformants and helps in growth of the transformant. Thereby it eliminates the non-transformant. That is the role of selectable markers. Next is the cloning sites. The vector needs the cloning sites. It should be a recognition site. They are mainly for the commonly used restriction enzymes. The cloning site, it has to be there. These are all the important features of a vector. Next is the types of vector. The first vector what we are going to learn is the plasmid. This is the widely used vector in genetic engineering process. This is actually the bacteria. This bacteria what we are seeing, it is called the E. coli bacteria, Escherichia coli bacteria. This is a prokaryotic organism. The prokaryotic organism, we can see that the nucleus are not covered by the nuclear membrane. The nucleus is separated. This is the nucleus, that is called as the nucleoid, or it is also known as incipient nucleus. So within the cytoplasm, apart from this nucleus, there is another DNA molecule, the circular DNA molecule. Can you identify this? This circular DNA molecule, it is called the plasmid. What is plasmid? Plasmid is an extra chromosomal material which have the ability to self-replicate. It is a circular DNA molecule. This is found in certain bacteria in addition to the genome of the bacteria. Plasmids contain genetic information on its own. They can replicate on its own. That is about the plasmid. This is important plasmid which are widely used in the field of biotechnology. This is called as the PBR 322 plasmid. Children, there are certain regions are here, right? So now I'll give you 5 minutes time. Just copy this slide. Just copy this alone. Possibly just copy this whole slide, PBR 322, this whole slide, that will indicate what are the different regions are present there in the PBR 322 plasmid. It is a very important plasmid and very important thing, you should know it. Just copy this slide. I'll give you 5 minutes time. Children, now we are going to talk about the vector, important vector PBr 322 plasmid. This is an PBr 322 plasmid. In few exams they asked to draw the diagram in board exams, and they also asked to find out the various regions that is present in this particular plasmid PBr 322. This is the reconstructed plasmid, and they are widely used as cloning vector. It contains 4361 base pairs, and here the PBr, the P denotes the plasmid, and B and R, it mentions the scientists, those who have identified or developed this plasmid. It's called Bolivar and Rodriguez. That's why it is called PBr plasmid. The 322 is the number of plasmid developed from their laboratory, and it contains the two important antibiotic resistant genes are there. That's called AMPR, TETR. The AMPR, it means ampicillin resistant gene, and TETR is called tetracycline resistant genes. These are all antibiotic resistant genes that are present there, and there are also a lot of recognition sites are there for certain enzymes, for EcoRI1, for HIND3, for BAMH1, and also SAL1, PUV2, PST1, CLA1, ORI1. ORI, that's called origin of replication. This is called origin of replication region, and antibiotic resistant genes also present here. And ROP, this is the protein involved that codes for a protein involved in replication of the plasmids. So this is the typical structure of PBr 322 plasmids. The other plasmid is called TA plasmid. This is called tumor inducing plasmid that is found in Agrobacterium tumefaciens. And this is now this particular TA plasmids are widely used in the field of genetic engineering. This is Agrobacterium tumefaciens. The bacteria, they are responsible for inducing tumor in several dicot plants, and this carries a gene that is called plasmid transfer. Plasmid carries TRA gene, which helps to transfer DNA from one bacterium to another bacterium or plant cell. That's why the TA plasmids are widely used in the field of genetic engineering process. And these are some of the regions that are present in the TA plasmid. We have the tDNA or TA plasmid. It is a stable integrated with plant DNA, and it contains the ORA regions is there, and also it has the ONC gene for oncogenicity, which causes cancer, or ORI gene, that is for region of origin of replication process, and INC gene is there for incompatibility. And this Agrobacterium plasmids, TA plasmids, are used to incorporate or introduce the genes into the plant's gene of desired traits into the plant. That's called TA plasmids. Others, transponents as vector. Here transponents are also used as vectors. What are transponents? The transposable element or mobile elements. Here these are all the DNA sequences able to insert itself into a new location without having any sequence relationship. The transponents, just like that, it can interpret into the DNA sequence. You can see there, this is the transponent. Just like that, it attaches to the DNA molecule. That's why the transponents are known as walking genes or jumping genes. Without any interrelations or without any sequence relationship, this transponents can easily attaches to the DNA molecule. That's why they are called walking genes or jumping genes. And they are all the genetic tools for analysis of genes as well as protein functions inside the host cells. The transponents are well studied, are used to study the Arabidopsis thaliana and bacteria, Escherichia coli bacteria. This is also used as vectors. And cosmid and pagimid, these are all the vectors. These are all other type of vectors, and the other vectors, the cosmid vectors. The cosmid vectors, it contains the cos site. That's called the cohesive terminus. The sequence having n for the cohesive end. And also this is an hybrid vector. This is a combination of lambda phage DNA vector as well as the cos site of bacterial plasmid. That's why this is called hybrid vectors. And pagimid vectors, this pagimid vector, this is again a reconstructed vector which contains the origin of replication region, and also you can see that amphicoelin genes are there, also contain this is also a page linked pagimid vectors are there. This is about the types of vectors. Next we are going to talk about the genetic engineering process, how exactly it is done. Previously I have explained you what is genetic engineering process. Genetic engineering process. Okay children, so the new topic, the genetic engineering, as well as we are going to talk about the tools of genetic engineering and the various methods of gene transfer. All these things we are going to talk about the next class. Now another 10 minutes is there. This 10 minutes I want you to have a discussion with me. If you have any doubt to be discussed, let us discuss that. And also I want you to draw this diagram, and also the other 10 minutes it is allotted for you to clear your doubts. So children, now you can ask your doubt.